essentially the past OIF experiments mapped on a world map. So Victor, please correct me if those locations, if there's some locations missing here. I know at least of one location in Canada that was not academic, but that created, I think, by quantity, quite a large iron input, I think 100 tons. And there's, I think, one paper published on it, but it didn't make it to the map.
You have shown all the iron fertilization experiments that have been carried out up to now. The one in the North Atlantic is not so much an iron fertilization experiment because I'll explain just now to you. And the experiments were carried out in zones that are called high nutrient, low chlorophyll zones. And these are places where we have upwelling of ocean water in the middle of the ocean. These are not the coastal upwelling zones, which border the western seaboards of the continents, but these are in the open ocean. The Southern Ocean is the best known of the high nutrient, low chlorophyll regions. I refer to them as HNLC in future, HNLC. And the three major HNLC regions are the Southern Ocean, which is by far the biggest, and the place where this would also work, iron fertilization to remove carbon dioxide, as it has in the past. And the others are along the equators. And there you can see from IronEx 1 and 2 in the Pacific. Those were on the equatorial upwelling. And then the other large HNLC region is in the North Pacific, where these three experiments, SOIREE and SEEDS 1 and 2, two experiments were carried out over there in successive years with different results. Both were carried out in the same location in successive years. All right. Yeah, just let's keep this map first. Now, the regions where nutrients come up in the open ocean are places of divergence, where water, surface water, is pushed away from coastal regions, but deep into the open ocean. So the North Pacific is one such region where you have a gyre, which is counterclockwise. And because it's counterclockwise, the northern hemisphere is pushing water outside. And that's replaced by upwelling in the middle of the gyre. And the Southern Ocean is also such a place. The equatorial Pacific, on the other hand, the Pacific upwelling is due to the Coriolis effect pushing along the equator to the north and to the south on both sides, right? So you have upwelling in the middle. So the equatorial waters are cooler than the waters north and south of the equator because of this effect. Now, the first experiment that was carried out was IronEx 1 in the equatorial Pacific. And there, what happened is that they had a rain event, a squall, they called it, right? They had very heavy rain in the vicinity of the patch after they had fertilized the patch and everything was they were already taking the first measurements. This rainwater created a low-density layer, right? And this low-density layer then overrode the patch, submerged it, so to speak, with a 30-meter layer of surface water from adjacent that had become lighter and therefore spread out. So they couldn't follow the experiment properly, but they had enough data from that first experiment to show that it had an effect and that the patch was retained. Actually, that patch made the cover a picture of Nature, the journal Nature, a couple of years later when they published it. So it was clear that effect was there, that the iron effect, that the iron addition had a massive effect on the phytoplankton. And that converted me. That was enough to convert me. And then I think 2 years later, they again got ship time to carry out IronEx 2, and they went a bit further out. And the problem with the equatorial upwelling is that it's upwelling, but it's also downwelling because it's adjacent. And then there are very strong currents. So there are very strong currents that flow to the west. And the patches in this IronEx 1 and 2, they were transported during the course of the experiment of just 2 to 3 weeks. They were transported like 1,000 kilometers, I believe, to the west. So they had to chase the patch. westward. Right. And this upwelling water along the equator also then goes down. And the problem here is that you might get a big bloom, you might pick up CO2, but before that CO2 deficit water is equilibrated with the atmosphere, it downwells again. So that means that you don't get anything out of it. You don't take up, there's no net uptake of CO2. And as a matter of fact, equatorial upwelling used to be, I'm not so sure what the status is now. The status now meaning with rising CO2 levels in the atmosphere. But let's say 20 years ago, it was the largest single annual source of CO2 from the natural ecosystem, from the natural world. And it was 1 gigaton a year CO2 was emitted because of the upwelling. This is cold water coming up. It's warm, so it gives off CO2 because it has an extra amount of CO2 in it, right? So now the CO2 level is higher in the atmosphere. It's less, but it's still a source of CO2. It has a lot of nutrients, but because there's very little iron in deep water in the ocean, it's just not enough to compensate the nitrogen and the phosphate that are brought to the surface. Okay. So after IronEx 1 and 2, the next experiment was SOIREE, which is south of Australia. That one, SOIREE, right? And SOIREE was carried out for 2 weeks, and then they had to leave the place. And they created a big bloom. And one of the features of this experiment was it was only 2 weeks. And the irony of it all is that this is a ship, an Australian research vessel, that got these 2 weeks to carry out this experiment, meaning probably a total of 1 month, 1 week to get there, 1 week to go back, and 2 weeks on site. And this SOIREE is a world-famous experiment because it was also on the cover page of Nature and a lot talked about and so on. But what the ship was doing before and after, nobody knows. Anyway, so SOIREE also had a kind of impact because after they left the site, 2 weeks after adding the iron, there was a satellite image of a crescent. They just went somewhere and dumped the iron, right? And the Southern Ocean has very strong currents, and these currents pull fronts, right? They have fronts which are going round and round. These are boundaries between water masses. And then what happened, they found a crescent, chlorophyll crescent on the satellite image. And it was where they had done the experiment. You could relate it to that experiment. This was interpreted as, oh my God, the long-lasting effects of iron fertilization. This was 1 month later, right? And who knows what this effect this is going to have, and all that, blah, blah. That came out of this experiment, that it has a longer effect than normal. And we solved all that with our experiments later on, the IronEx, EIFEX, and LOHAFEX experiments in the Atlantic. And we showed that this is all an effect of the biology of the dominant phytoplankton that are running the thing, right? So in SOIREE, they did look at under the microscope, and there was a species of alga, which is perhaps the commonest species in the Southern Ocean. Has a very long name, Fragilariopsis kerguelensis, but it has a major impact on the globe, this particular species, for various reasons I can explain to you later on. So I have been looking at this species for quite some time, and I can explain why the SOIREE bloom was still there 4 weeks later. Okay, it was not studied. It was just appeared on a satellite image. But I'm pretty sure if we had done a local study, we would have shown that this is Fragilariopsis responsible for it. And as I said, it's the most common diatom in the Southern Ocean. And it's not only in the surface water, it's also in the sediments. It makes very thick sediment layers. All right. So then we move on to SOFeX. These were 2 experiments in the Pacific, in the Southern Ocean Pacific. And they also did not look at the species. They carried out these experiments. It was on 1 cruise, just to show you, right? So the same research vessel that goes out all the time did this experiment on the side. Never before, never after, because then other research programs were deemed more interesting or more important or whatever. And that pains me to think of how we are squandering not only our money, but also our research time and scientists' time and so on. Okay, the SOFeX results, they both led to big blooms, and they were not so evident on satellite images because the Southern Ocean is very cloudy. It's a very windswept and very violent kind of ocean, the most violent, right? The roaring 40s, famous, and the furious 50s, right? And I have spent a lot of time over there, so I really know the place very well. And now there were also 2 experiments carried out in the North Pacific, SEREES and SEEDS 1 and 2. SEREES was carried out by a team from Canada. And one of the authors, one of the leading scientists on board SEREES was also the person who had done SOIREE in New Zealand. Actually, I'm not so sure which came first, SEREES or SOIREE. It was about the same time in the late 90s. So what they found in SEREES was actually an absolute classic diatom bloom. They had a big bloom. I think the experiment lasted 4 weeks or so. And they had a mixed bloom of Chaetoceros and Thalassocyra. These are classic coastal species. But this experiment was carried out in the open HNLC area of the North Pacific. It just shows you that these species are also practically, they can be induced to grow everywhere. Right? Now, in SEREES, what happened was that they had this big bloom, and then after 3 or 4 weeks, it vanished. And the problem there was that the water below that patch was monitored, but that does not mean that it stayed congruent to the fertilized patch on the surface. It's very likely that it went in a different direction because these water masses are not necessarily, the deeper water masses are not necessarily connected to the surface water masses. So my hunch is that bloom also sank out, the SEREES bloom. But the people at that time, and I also heard there was a lot of controversy, they preferred to say that bloom biomass was broken down in the surface layer. And I think that's a huge mistake because it's impossible to break down bloom biomass so rapidly. And the most obvious explanation is that it sank out. But I don't know why, but perhaps political reasons and so on, they preferred not to say it sank out because then the venture capitalists would have said, okay, we are going to do this in a big way. So they preferred to say it didn't sink out. And now I think they are the people who were involved. They're saying something else, that it did perhaps sink out and they didn't record it sinking out because that water layer into which the bloom sank moved away from under the patch, which is most likely what happened. That SEREES experiment was given a big question mark, like probably didn't work. Although I'm pretty sure that it did, if they had done a proper hydrographical study. Now, SEEDS 1 and 2, these experiments were run by Japanese scientists, and they only had, I think, 10 days for SEEDS 1, a very short period of time. And what happened during SEEDS 1 is that they had a massive bloom of Chaetoceros debile, which is very similar to what's behind your head. And it was like, I think, 80–90% or so of the biomass was this one species. But then again, here, like SOIREE, they had to leave. And the North Pacific is actually a calmer ocean than the Southern Ocean. So the mixed layer is also shallower. So the mixed layer was only, I think, 10 meters deep in SEREES 1. But as I said, they had to leave. And I'm absolutely sure, 100% sure, I would absolutely bet anything that it sank out in the weeks after that. 2 or 3 weeks later, there must have been a massive outfall of that Chaetoceros bloom that would have gone down to the seafloor. Absolutely sure. But they were not able to record it because they had to leave.
Do you feel it's fair to say that you have the only published peer-reviewed experiment that proved carbon export, but the other teams didn't actually have enough time to prove carbon export or didn't have the experiment to set up? So that does not mean that it doesn't work for export. It just says people haven't tested for it.
Yeah, the experimental setup wasn't correct. It was too short. You can't have a diatom bloom in a couple of days and have it sink out in a couple of days. These things take their time. In the equator, where the water is much warmer, it's not as warm as it is in the subtropical gyres, like 30 degrees nowadays. but it's still 20, 25 degrees, right? You know that from the Galapagos Islands. The Galapagos Islands also there. The water there is cold, cool.
And to flip it on the head, were there any experiments that had the right experimental setup that could not show any sinking out or any carbon export?
No. That's why I'm absolutely convinced. No, this is normal. This is what I know from my years of studying natural ecosystems, how diatom blooms behave. I know that. And if, it's like having a tree, and in July, oh, this is not a deciduous tree, it still has leaves on it in July, in the middle of the summer, right? So that's the level at which this discussion is taking place, which is painful because it's so against what we know and appreciate. Not we, okay, I'll say I, because I can't help it, but I just have much more experience with plankton than very many of my colleagues, particularly those who were involved in these experiments. Now, to come back to the Japanese experiment, seeds too, and this is, this is the exception. You're right. There is one exception. Seeds 2 was that exception. What happened in seeds 2 was that they repeated seeds 1 and they had more time there. It was an excellent study. They did a very good job of studying the plankton, describing it. But there was no big diatom bloom there. And I can't explain that. But all I can say is that this particular diatom, the one that made the bloom in seeds 1, was not present for whatever reason. It just wasn't there. So that bloom didn't develop, and they had a kind of normal plankton increase in biomass and activity and so on. But there was none of that boom and bust situation that you get in bloom. So this was an exception. And it would be interesting to find out why there was no diatom bloom, because there was enough silica.
There's one more experiment that is a controversial one, but it created a bloom and it was in the same region. And the creator of this bloom, the creator of this experiment, claimed that the next year or the 2 years afterward, there was a huge amount of salmon. So much larger than usual, and it helped the local fishery. And you just mentioned that this area might not be good for carbon sinking, but it might be good to restore our ocean food webs.
Absolutely. I'm convinced about that because we also had the same whale diatom krill situation in the North Pacific. After the Southern Ocean whales were wiped out, the whaling vessels went north between Kamchatka and Alaska and wiped out the whales that were there. And that region, as I said, is also a high nutrient, low chlorophyll region. And the whales were fertilizing the krill. I'm sure that the same thing happened there. And the same with the salmon, that there are years where there are much more salmon than in other years. And the problem with that is that people haven't studied this properly. So my gut feeling is that salmon, on the one hand, the growth rate is limited by the availability of food. So how fast, how many years they take to reach adulthood is important. But the other thing that is also important is that these salmon, before they go upriver, they all congregate at the river mouths, right? And if they haven't eaten enough that year, if there's not enough zooplankton, then they just don't have the fat and the energy to swim up the rivers, and then they don't swim up the rivers, right? So if you have years where you have a much bigger influx of salmon in the rivers, it doesn't mean that those salmon are from the same year class necessarily. It could also mean that they just had more food in front of the rivers at that time, which is why, again, you don't have the thing across the entire Pacific. All right. I'm sorry, I haven't read the literature in detail, and I'm sure that the Canadian colleagues have done work on this, but I would like to know what is the nutritional status of salmon in front of the rivers from year to year, and how does it vary, and how does that relate to the food available over there at that time, right? Because they're coming from the entire HNLC area, and then they congregate in front of the river mouths, and they have to be in good condition. If they do this with bad condition, there's no problem. So they're not going to lay many eggs. Those eggs are not going to be viable young, and so on and so forth. So you have to look again at the life cycle of the salmon in this case, right? Because the Canadian government has been growing fish larvae, salmon. They've been collecting the eggs and growing them in hatcheries and releasing them. And that has had no effect on the salmon.
So the interesting part of this experiment was that the quantity of iron input was much larger by an order of magnitude than the other experiments. So according to my knowledge, they put in 100 tons of iron. And last time we calculated that this may very well created 50,000 tons or maybe even more of phytoplankton biomass. Depends what phytoplankton was growing there before.
I would be careful there. I mean, I don't think we can even try to estimate how much phytoplankton biomass. Let's say that there were studies done by people who looked at the zooplankton. They were independent of the experimenters. There's a routine program where what's known as a plankton recorder is towed behind a passenger vessel, and it records the plankton on that stretch. So there's one ferry that goes to Alaska. Anyway, it goes to that area. And they checked the zooplankton on this plankton recorder from that period, and they had more zooplankton. So this iron fertilization experiment is called a rogue experiment, right? It should have been done by scientists long ago. Anyway, it was not. So this entrepreneur who did it, who convinced a lot of people to join him on this. There were hardly any bona fide scientists on board. But as I said, this other crews showed that there was more zooplankton, which would mean that the salmon there had more to eat. And that might be the explanation for the better year class, because it was too short for it to have an effect on the population size, right? But the quality or the status of the migrating fish could have been dependent on that. And there are also papers of a volcanic outburst on the Aleutian Islands, where they had a huge salmon run in a year or two after that. I think, the year after that. So that would be an indication of increasing the biomass of the salmon, right? So there was just simply more salmon.
Yeah, in fact, we spoke to one scientist, not the founder of the company, not the entrepreneur, but we spoke to one scientist that he hired, who was basically leading these experiments from a scientific point of view. And he told us that within hours, they could see the phytoplankton growth. They could see that there were more birds, that there was more fish activity. They even saw whales. And he believes, when he talked about this experiment, and this is a scientist speaking, his eyes was lightening up, and he was so excited about how this biosphere started getting active again. So do you think it's possible to fertilize the ocean with iron, and then within, let's say you are on a cruise ship and this is like an attraction for a cruise ship, and the cruise ship provider would put a ton of iron, would the guests on the cruise ship be able to see this phytoplankton bloom within 1 hour? And would they be able to see the whales within days? Or how quickly could it work down the chain? Or was it just a coincidence that they saw so many birds?
I think that this would happen pretty quickly. Yeah. And we've seen that in the Southern Ocean, that within hours, the phytoplankton physiological growth, not what you would see in terms of biomass, but the physiology of the phytoplankton improved within hours. We were circling. We made our patch by circling like that, right? So we gave the positions to the captain, and then a 1 kilometer distance. And while we were doing this, we were monitoring the physiology of the phytoplankton. So we were doing this, and suddenly there was one place where the phytoplankton was much, much more active. And what happened over there, definitely, is that we had cut a previous circle made by the ship. So that phytoplankton had already improved its physiology within hours. And with regard to the experience I also spoke to this person who was on board that experiment. He told me the same story. And I don't see reason to doubt it, because I know from the Equatorial experiment, when they carried out the Equatorial experiment, that they had the same thing. The place just exploded. There were fish landed up there and birds and so on. And then you wonder how they would have got the information, because the water itself is very slow. It's not spreading the stuff. So they got it from the air. The phytoplankton is giving off molecules like DMS. This is this dimethyl sulfide, there's another story again that I can talk about for another hour if you want. But this DMS is smelled by birds. They've done that experiment, and birds smell that. And we can also smell it. It smells what you smell on beaches in the summer. This is dimethyl sulfide. It's not pleasant, but you wouldn't use it as a perfume. It's not like H2S. It's not a stench, but it's the smell of the sea, the summer ocean. And that birds pick up and they're attracted to it because what it means is that there's this phytoplankton bloom that give off this DMS. So it's already there in place. So I believe this guy when he says that the birds also were attracted. And the whales, of course, are also coming up and breathing the air, right? And they also, most probably, this has not been shown yet, but I'm sure they're also smelling DMS. Yeah, so for cruise ship people, this would be interesting. We could see how long it would take. Now, the interesting thing is that in the Equatorial Pacific, when they carried out this experiment and they had this response, instead of getting excited and saying, oh, wow, we humans now have a lever to manipulate the ocean in the favor of the ocean, they got shocked. They were worried. They said, oh my God, what have we done? And scared. And this is an attitude a lot of scientists, unfortunately, have. That instead of being forward-looking and seeing what we can do and learn and how we can apply the knowledge that we learn, they get scared and say, oh, we've done God knows what, like with the SOIREE bloom that was lasted for 4 weeks. Right? And the other belts, they don't get anything. And I didn't mention this experiment that was carried out south of Australia. It was in that map you showed in the beginning. I've forgotten the name. It was a very short experiment, but they also showed that there was an increase in phytoplankton. It was very short. But LOHAFEX was a long experiment. It lasted for a month and a half. And there we showed that we got a big bloom. It's not a huge bloom. There was no silica for LOHAFEX. There was no silica, extremely low values. And there was no diatom bloom. And we very closely monitored sinking from that bloom, and we could show that very little sank really to depth. It was all retained in the upper layer, the upper couple of 100-meter layer. So there was no fallout. And that was the function of, or that was the purpose of LOHAFEX, to show where it doesn't work, where you don't have silica. And you don't have silica there because it's been taken up by diatoms that have a very...
But you have low silica, right? You don't have no silica, you just have this.
So this is no silica. So there's no point in saying, all right, there's low silica, but we'll still get something. Yeah, there were diatoms there. The diatoms grew, but they were at very low levels. Just because they didn't have enough silica to make enough cell walls for enough.
So there must be low silica. So there must be some silica there, otherwise they couldn't grow.
Yeah, there's always some silica there, but that has no effect then on the carbon sequestration.